Fantastic. So thank you very much for the introduction. I want to talk about biological application of super resolution microscopy. So we've seen a lot of talks about fantastic developments, and this should give a glimpse into why we're really interested in those developments. Now, we've been working for quite some time on studying the nuclear interior, nuclear organization by optimizing 3D structured illumination microscopy and get ever more detailed 3D super resolution images. And we've also developed quantitative workflows to get really meaningful numbers from our data. And more recently, we ventured into electron microscopy, more specifically FIB-SEM. So just to sort of set the stage, if you look into the textbook, of course, and look into the organization of chromatin, you will see the organization of the DNA around histone proteins to form nucleosomes. They form a bead on a string conformation. Importantly, we have histone tails that carry post-translational modification that carry information about, for instance, transcriptional activity or repression. But then on the higher level organization, often textbooks end up in this sort of 30 nanometer fiber ending up as a spaghetti in the nucleus. And what we now know very much is that all these views are pretty much wrong, and there's a much more complicated arrangement of chromatin. That is still really ongoing research. So the really important, very fundamental question in the field is how chromatin is organized really at the mesoscale, so in this area in single cells, and how this structure affects nuclear function and vice versa. So one really big problem is how to image chromatin in cells. And if you look, go into the internet and look at, highest resolving EM data, you find images like this, and you see these darker areas within the nucleus that are annotated as heterchromatin, these brighter areas that are annotated euchromatin, the nucleolus showing up very, very prominently. But then you can look at other images, EM images, and suddenly you do not see any of these heterochromatin regions, but rather very kind of evenly fuzzy image of chromatin. So you can see already that dependent on the preparation method and the way of imaging, you can get very different outcomes. When we look at optical imaging, of course, we have the problem of limited resolution, and particularly when it comes to imaging DNA or chromatin, this very dense and thick organelle that creates a lot of out of focus blur and very little contrast. So by using structured illumination, we can improve on this problem or get some solutions to that. And I know we've been talking about this a lot, but we exploit the Morée effect and the ability to rescue or shift high frequency information into an observable region by putting a structured illumination onto our samples, and then by recording a series of raw data with shifted stripes and rotating stripes, we can recover high frequency information from all directions. And in that way, and then by reconstructing this data, we can really get a much more detailed view of our structures, of our complex structures, with eightfold extended frequency volume, but also really a much higher contrasted image. And this is really important. So we basically get rid of all the autofocus blur. And that allows us to look at this very complex structure in a meaningful way. Of course, we are always mindful of potential artifacts of SIM, and therefore we developed the software tool SIMcheck that allows us to quantitatively assess the underlying modulation contrast that really affects the presence of artifacts or not. So this looks, that basically tells us that our structures that we see are really reliable and real. And we can also assess quantitatively the resolution in our image, not by just doing some selectively choosing some structures and doing full width of half max measurement, but really looking into the overall frequency range. And we can very nicely see, for instance, in this case, that we have a resolution in our data of about 110 nanometer in a lateral direction and maybe 300 nanometer in axial direction. So with this technique, very early on, we've been identifying these larger domains of chromatin. It looked like filaments or higher order domains. And in between there is apparently DNA-free space, which we termed interchromatin space, and sort of channels that then lead to nuclear pores. And because we image really in 3D space, we can then segment chromatin and interchromatin and assess this three-dimensional topography very, very nicely. Now, one first major question, of course, was, okay, if there is this interchromatin space, what do this interchromatin space harbors? What is this filled with? It's apparently not empty. So a very revealing experiment was when we dual labeled DNA with bulk RNA by a long-term EU pulse labeling of bulk chromatin. And if you look at both labels, essentially we have the second side of our coin. It's pretty much the inverted image of our chromatin landscape. So we have really two almost exclusive compartments here. And if we just look at the distribution of and the sizes of these domains, they're around 200 to 300 nanometer wide. It's important to note that most of the RNA in the nucleus is not messenger RNA, but non-coding RNA, so ribosomal RNA, rRNA, and tRNAs. How about dynamics? So we basically try to optimize our 3D imaging to do time-lapse imaging. And these are the kind of data that we see. And I think it's clear from these data that we see the same domain organization also in live cells, so we're not looking into fixation artifacts, and we have some dynamic properties that we can assess also quantitatively, and don't want to dwell too much on these analysis, this flow field analysis, but essentially what they told us is that we are really looking here at coherently moving mechanically coupled domains with viscoelastic properties. So essentially we're looking here at blobs by definition, not only because of our resolution. But of course, common criticism is, okay, SIM, despite being super resolution, it's still diffraction limited, and therefore we still only have 100 nanometer. Everything will look like blobs. So we had to find a way to look into greater details into our structures, and teamed up with Harald Hess from Janelia, who at the time developed a much advanced focused ion beam scanning electron microscopy approach, producing incredible detailed three-dimensional EM data of the same cells that we have been using. So in this case, Hela cells. And if you look into details, so this is basically an orthogonal, a physical section where the surface is scanned by scanning electron microscopy. And here you really nicely see these assemblies of nucleosomes. And I should say these cells have been cryo-fixed before preparation for EM. So the fixation protocol is really, really structure preserving. And you can see here really nicely these assemblies of nucleosomes forming these sort of domains, and these are associated with the nuclear envelope, but also inside the nucleus. And this is very, very, very reminiscent of what we've previously described with 3D SIM. So we're looking at really at the same kind of structures here. And because we look here, we have a full 3D set, we can basically analyze this through the entire volume of the nucleus. Here you can see, and I'm showing some more insets, this is the nucleolus, and inside the nucleolus we have all these isolated black dots, and these are ribonucleoparticles. And we know that ribonucleoparticles also kind of travel through the nucleus, and we think that most of the isolated dark spots here outside these domains are also ribonucleoparticles. But then we have these what we call nanodomains, and they are often seen to be linked via sort of smaller filaments into chains of nanodomains. I'm going to skip this, and I just want to say that we've established machine learning assisted segmentation to segment these domains and also these isolated dots, and basically kind of reproduced the resolution that we achieve with 3D SIM, so by a sort of blurring and projecting enough set stacks, and essentially we can very much regenerate the same images that we obtain with 3D SIM. So we think we have a very nice correlation here that allows us really to segment in 3D and follow the network of domains. So as a final step, we've been trying to generate a functional map on top of our nuclear topography by mapping a whole set of nuclear markers and functional markers onto this 3D landscape. Basically, we applied a specific segmentation procedure that allows us to define different chromatin classes that basically act as a proxy of inside or outside. So the interchromatin space being class 1, and the peripheries here class 2 and 3, and so forth. The interiors of these domains being in these higher classes. We established a quite elaborate workflow of segmentation and annotation and mapping onto these classes. But essentially, what we produce here are frequency maps of relative enrichment and depletion of nuclear markers, where we can very nicely define X, Y, and Z coordinates onto this intensity classes, and basically look at enrichments of markers and depletions, and also distances to segmented boundaries. And this is, I'm really rushing through here because there's a lot of biology in here. But for instance, we looked at cohesin, a main organizer of chromatin, involved in loop extrusion, and we can see that this is very much located at the periphery of these chromatin networks. H3K4, and marker for active promoters, also very much peripheral apparently. But when we look then at more repressive markers, they are much more internalized into these domains. So we can visualize this with these kinds of heat maps, and each heat map or each section here are 20 complete, like full volumes, 3D volumes, 3-color, 4-color, imaged, analyzed, and only one particular cell cycle stage, G1. And basically what we see here is that all RNA-associated factors that we look at are very much enriched at the periphery and the interchromatin space. Similar CTCF and cohesin. But then when we look at active genes, they are pretty confined to the surface of chromatin domains, of course depleted in interchromatin, but also at the interior regions. Skip this, and silence genes then very much, of course, shifted towards the interior, and yeah, that's also reflected in this very heterochromatic markers. So what does this tell us? So we basically have a functional topography with active chromatin surfaces and repressed cores. And that is very much correlates with the sizes of the enzyme machineries that sets these markers. So those enzyme complexes that are working basically or associated with markers at the exterior, at the outside, are also the largest ones, whereas the ones in the interior markers are the smallest complexes. So there seems to be really a strong size correlation and an exclusion effect. So we've used this workflow to look at various biological processes, from DNA replication, loop extrusion, senescence, DNA repair, you name it. And I want to refer to those papers if you're more interested into that. But what I want to really, the message I want to take home today is that, we have these domains, and really the active part, the euchromatic part, if you like, is, instead of being here, and this would be heterochromatic, is really euchromatin is at the surfaces of domains, heterochromatin or silence chromatin in the interior of these domains. And we have a separated compartment that is for RNA processing. So in a way, it's a new paradigm how to think about euchromatin and heterochromatin. So that are basically the models that we derive from our data. Don't want to go into detail, just summarizing that chromatin arranges into mesoscale nucleosomal aggregates or blobs that are separated, juxtaposed to RNA-occupied interchromatin space. And it's, of course, tempting to speculate that this is an effect of phase separation and charge of nucleosomes and effects of ATP-driven machineries like transcription, replication. Activity is strongly confined to CD surfaces, and repressed chromatin is enriched in the interior. What I haven't shown, but I'm just telling here, that cohesin is dispensable for this sort of topography. So it has other roles, but not to set up this kind of topography. And DNA replication locally disrupts this topography, creating a window of opportunity to access hidden sequences. So with this, I want to thank my co-workers, particularly Ezekiel Miron, who did most of this work on this particular project, as well as Roel, a former master's student. And of course, nothing couldn't have done this without the excellent collaboration with Micron, Oxford, and others in the department. So with this, I want to thank you and happy to take questions.
Okay, thank you very much, Lothar. That's really interesting work, and it's quite closely related to some of the stuff we're doing in one of the projects we're involved with in Imperial at the moment. So I'm very interested to see the results you've got so far. While we can wait and see if anybody has any questions in the chat, but I was going to ask, can you tell us anything about the hardware you're using for your?
Yes, I can. So far we've been relying on an OMX microscope with the Blaze unit, so a commercial setup. We've been collaborating with the company at that time, GE or API, to optimize some of the parts in there, but it's pretty much a commercial instrument, and optimizing the imaging as much as possible to avoid any artifacts.
Yeah, you seem to be particularly low on artifacts. I thought, your images looked particularly clear there.